So today we are going to discuss the last chapters for your sixth semester students. That is gene transfer technology in animals, and specifically it is mentioned that transferring of the genes into the animal oocyte, embryos, and specific animal tissues. Okay. Now, what is a transgenesis? Before going to transgenic animals, what is transgenesis? Transgenesis may be defined as the introductions of exogenous foreign DNA into the genome such that it is stably maintained in heritable manner. Animals that have been permanently engineered by gene insertions are called transgenic animals, and any foreign gene that are added are called transgenes. Genetically modified, genetically engineered, and transgenic organisms are often used interchangeably, yet they do not mean the same things. A genetically modified organism is one that has had its genetic material altered through any method, including conventional breedings, cross between the animal beings, and that is known as the genetically modified organisms. Okay. Now, a genetically engineered organism is one that has been genetically modified using recombinant DNA technology. Another variety, that is a transgenic organisms, but transgenic animals has been genetically engineered using a foreign gene usually belonging to a different species. Now, the first step of transgenic animals in DNA recombinant technology. Because before going to transgenic animals, we must know the recombination technology, how to recombine. Okay, so that already I have discussed in genetic engineering and genetic engineering. But still, again we are recollecting some information. DNA must be manipulated when transgenic animals are created. DNA is altered using the genetic engineering method called the DNA recombination technology. Transgenic methodology has already been applied to diverse organisms, for example, mice, fish, and microorganisms such as fungi and bacteria. This technology involves ligating the DNA fragment, encoding the transgenes into a vehicle such as plasmid vector, which is used to amplify the DNA. A plasmid is a small circular piece of the DNA found in the bacteria, which can replicate quickly in the cytoplasm to make a large number of the copies of the cloned DNA. The cloned DNA is then purified from the bacteria and inserted into the host, that is the mouse or maybe other animal beings. Now, this is a pictures how to prepare, that is a recombinant DNA technology. So here in this picture I have shown the one plasmid which is collected from the bacteria. Plasmid DNA is taken, and a foreign DNA which consists of the particular gene, maybe hemoglobin, maybe lactalbumin, maybe insulin. Okay, the foreign genes. Now foreign gene is cut and the plasmid DNA is cut with the help of the restriction enzymes, eco R1 restriction enzyme. As a result, the staggered cut is prepared. Now this two staggered cut is again ligated by annealed with the help of the ligating enzymes. Okay. As a result, there is the formation of the recombinant DNA or chimera DNA. Okay, and which is the first and foremost requirement for what transgenic animals. Okay. Next, let us discuss the details of the transgenic animal. What is a transgenic animals? Now, the genetic characteristics of the livestocks and other domestic animals in the early days was carried out by selective breeding methods. This technique primarily involves combination of the mating and selection of the animals with improved genetic traits. The selective breeding is very time consuming and costly. It was the only method available till some years ago to enhance the genetic characteristics of the animal. So this is the most earliest methods of crossbreeding experiments. Okay, but with the help of the introduction of the biotechnology, it is now possible to carry out manipulation of the genetic level to get desired characteristics in animals. A transgenic animal is one that carries a foreign gene that has been deliberately inserted into a genome. Transgenesis is the process by which mixing of the gene takes place. Foreign gene is inserted into the germline of the animal, so it can be transmitted to the progeny of offsprings. The transgenic technology led to the development of fish, livestock, and other animals with an altered genetic profile, which are useful to mankind. So, my next slides. Transgenic refers to the phenomena of introductions of exogenous DNA into the genome to create and maintain stable heritable characters, high yield, high milk yield, more laying of the eggs, etc. So these characters. Once it is taken, the gene is taken, it will heritables, it will that is offspring after offspring, it will inherit. Okay. The foreign DNA that is introduced is called the transgenes, and the animals whose genomes altered by adding one or more transgenes is said to be transgenic. The first transgenic animal was the super mouse, created by Raphael Winston and Richard Palmer in 1982. It was created by inserting the human growth hormone gene in mouse genome. The offspring was much larger than the parents. Transgenic animals are important for improving the quality and quantity of the milk, meat, egg, wool production, B.Sc and drug resistance animals. Now, so in these two slides I have given the what introduction of the what doing the transgenic animals. Now in the next slides, how we will proceed. That is the method of creation of transgenic animal. How we will proceed. So microengineering, there is a four methods mainly. There is so many methods are there, but main methods of the four methods are there. Microinjection of DNA into the male pronucleus of the fertilized egg. Second, micro injection of the embryonic stem cell transfer to blastocyst. And gene transfer by using virus or retrovirus transductions and somatic cell nuclear transfers, clonings. So there is a four methods are there for what creation of transgenic animals. Now, before going to these macro injections, let us discuss something regarding the basic structures. Okay. So, prepare the transgenic animal, we must require the following objectives. We must have the following aims. What is the aim? First, gene of interest, foreign gene. Which genes we are going to introduce? That is the foreign gene of interest. That is not the foreign genes. Maybe insulin gene, maybe lactate, maybe hemoglobin genes. Okay. So, gene of interest, first criteria. Okay. Second, gene to be substituted by the foreign gene. So this foreign gene will be substituted by the foreign genes. So one gene will be replaced by these foreign genes. Okay. So that we have to first that is we must recognize that one, and then we substitute with the help of these foreign genes. Okay. Now after that, the desired genes should undergo recombination by different steps. For screening, we can run a southern blotting, ELISA, PCR, etc. Okay. We can find out whether the DNA is inserted or not. Okay. So this is the first and foremost criteria or aim of the transgenic animals. Okay. Another structure is what? When we are going to prepare the transgenic animals, okay, we must know these three terms. One is gene knockout, okay. One is gene knockdown, and one is gene knock-in. Now, genes knockout means genes is completely excluded, is completely removed. Okay, gene knockout. Gene we can artificially we can what gene we can completely we can block it. Okay, completely it is excluded. Okay, gene knockdown, blocking the expression of the desired genes. Gene will be there, but we can block the expression of the desired genes. Okay, it cannot synthesize the protein. So this is the gene knockdown. Another is gene knock-in, inserting a segment of the genes. Okay, additional genes we are inserting. So that is the gene knock-in. So this is one structural framework for what these transgenic animals. So in my next slides, first we are going to discuss the first method, that is which is not the microinjection of the DNA into the male pronucleus. Now, what is a male pronucleus? When a zygote, there is a sperm and there is a egg or ova fertilize, okay, it will give rise to a zygote. So in this zygote, before fusion of these sperm and there is a egg, there is a two pronucleus is found. One is egg pronucleus and there is a sperm pronucleus. Now out of the two pronucleus, the male pronucleus is more larger, the sperm pronucleus is more larger. There is a most of the transgenic technique is applied in the male pronucleus. That is a popularly known as microinjection of the DNA into the male pronucleus. Okay, and then other species it is applied first in the mice by Watson in 2000s, Watson 2000, and then it is other species applied in other species such as rat, sheep, goat, cow, pig, bird, chicken, and the fish. A pronucleus is the nucleus of the sperm cell or the egg cells. Okay, before they fuse together to become a fertilized zygote. But male pronucleus is larger than the female, that is why it is used. Okay. In this method, scientists clone the DNA into a vector such as the plasmid, then harvest newly fertilized egg before the pronucleus is fused. Next, the DNA is injected into the male pronucleus using the microsyringe. After that, the two pronuclei fuse to form the new zygote. Okay. After 5 days of the growth and divisions, the embryo is classified as a blastocyst. Okay, a hollow ball of cell, which is popularly known as the morula or a blastula. Okay, first is morula and then blastula, and where is a cavity is formed, then is the blastocyst. Okay, now This blastocyst now it is implanted into a pseudo-pregnant foster mothers, okay, prepared to receive an embryo by mating with the B.Sc animals. This is implanted into the pregnant foster mothers. As a result, what will happen? It will develop. So this type of the there is formation of there is a species with the help of the foreign gene. Introduction of the foreign gene is known as the microinjection of the DNA into the male nucleus. This is one kind of transactions. Okay. Second, so I have given one diagram here. So there is a diagram method 1 and method 2. In the method just you can it has been focused. There is a two pronucleus there. Out of that pronucleus, one pronucleus desired genes with the vector it is inserted. Okay, then it is fertilized egg. It is what if it grows, it will give rise to a blastula. Okay, and that blastula ultimately it is implanted into the foster mothers. As a result of which the offspring will be formed. Okay, and second method is also given here. The method 1 is given, though it is 1 and 2. It is not okay. First method I have discussed, that is the pronuclear method, and second method by the embryonic stem cells. Now embryonic stem cells, first it is collected. Embryonic stem cells, it is collected. Okay, then this embryonic stem cells. Okay, selection of the cell expression desired genes. Okay, cell is selected, and then this inject the transformation embryonic stem cell into the inner cell mass. Okay, another blastocyst. Okay, it is injected the inner cell mass and the blastocyst selected cell, selected gene. Okay, and then it is again implanted into the uterus of a pseudo-pregnant mothers by foster mothers. As a result, it will give rise to offspring. Okay, so this is very viable and percentage is very high in case of the mice, but in other animal beings, the percentage is very low. Next, how the microinjection is done. Just I have given you one diagrams so that you can understand very easily. A female animal is superovulated and the eggs are collected. Okay, the eggs are fertilized in vitro. Okay, the transgene containing the solution is injected into the male pronucleus using a micropipette. Okay, next the eggs with the transgenes are kept overnight in an incubator to develop in the two stages, ultimately morula, then the blastula stage. Okay, the eggs are then implanted into the uterus of the pseudo-pregnant female, that is mothers. Okay, female which has been mated with the vasectomized male and the previous nights. So in that we are learned microinjections of the DNA. There is it is done. Okay. Next is microinjection of DNA into embryonic cells. First is the pronucleus and then is embryonic cells. Now we see the second method. A second technique for making the transgenic animal is the incorporation of the transgenes, okay, into the embryonic stem cells. Okay, embryonic stem cells are inoculated, isolated from the inner cell mass of the blastocyst. First is isolated. They are undifferentiated cells. Okay, they still they are very young stage, undifferentiated cell. They are not give rise to a there is a ectoderm, mesoderm, and mesoderm. That is the undifferentiated cells. So have not yet generated structures and manufactured the protein characteristics of specialized type. They can renew themselves, okay, and can differentiate to produce almost all major specialized cell types. Okay, the process begins by collecting these embryonic cells are now collected first. Okay, the process begins by collecting embryonic cell from the blastocyst of the host, that is the mice. Okay, now a transgene is inserted in the cultured embryonic cells. Okay, here the blastocyst, it is again what transgene is inserted with the using the microinjection or electric treatments or chemical transfections, okay, or by viruses. Okay, then the embryonic cells are again injected in the blastocyst, injected into the blastocyst. As a result, what will happen? The blastocyst containing the transgene is now implanted in the pseudo-pregnant mother or the foster mother. As a result, what will happen? Transgenic animals will be developed. So this is the second method for transgenic method. Already slide already I have shown earlier. So here a picture is shown regarding the how there is cleavage. After cleavage, there is the blastula. In the blastula, there is a inner cell mass formation takes place. Inner cell mass is collected. Okay. Now inner cell mass cell is dissociated. Now in this dissociated mass, gene is introduced. Established embryonic stem cell is formed, and thus established embryonic stem cell now we can again incorporate into a another blastula, and thereby transgenic animals are produced. Now here in the next pictures, slides, it is nicely shown the how the there is a transgene is collected from the embryonic stem cells, and again after collection, now generation of the gene targeted mice, how it is implanted into the blastula, and after that, and the blastula is again implanted into the mothers, okay, foster mother, and mother which will give rise to ultimately give birth to the chimeric rats. Chimeric means it is a containing what strain genes which is present in the chimeric rats. When mating between the chimeric mouse and the normal mouse, okay, again they will give rise to the two types of the mouse, chimeric mouse as well as the normal mouse. Okay, so this is what a diagram showing regarding the how there is a embryonic cells, okay, gene embryonic stem cell helps in the what formation of the transgenic animals. So in my next slides, I would like to show the viruses. Okay, DNA and virus delivery. Viruses can also be used to deliver the clone transgenes into the embryonic stem cells. In this method, genetic engineering is used to insert the transgene into the place of a specific viral genes. Okay, viral genes is very limited viral genomes. Okay, in that we can very small gene also we can insert viral genes. Next, the viral DNA is then enclosed into the virion, and the virion is used for infect the embryonic cells. Okay, though effective, viral delivery is not efficient as desired because the size of the transgene sequence that can be added to the viral genome is very limited. Okay, also viral delivery can create a mosaic animals, and interference with the expression of the transgene may also occurs. Another problem that occur in the animal that are created will pass to the transgene to offsprings only if the germ cell receive the copy of the transgene, which does not always happen with these methods. So this is the DNA viral delivery, or it is also known as retrovirals methods. Okay, so here in my next slide I have given a diagram. So first a plasmid is collected from a bacteria. Okay, then in this plasmid there is a foreign gene is inserted. Red one is a foreign gene. The foreign gene is inserted. Then it will become the recombinant DNA. Okay, this recombinant DNA now cultured. Okay, after a few cells take up the DNA and produce the recombinant virus particles. Okay, do you follow my point? So there is a recombinant retrovirus formation which takes place, and this retrovirus containing the foreign genes allow to attack what another animal beings. Okay, as a result, what will happen? This retrovirus, when this DNA enters, okay, in animal being, so in that DNA foreign gene is also there. So that foreign gene ultimately it will give rise to the our required proteins. So in that way, what happens? Transgenic animals it can be prepared. Okay, or transgene can be incorporated into the another animals. Okay, this is the third method, and the last method that is known as that is somatic cell nuclear transfer. This is the most important, and this is also viable in case of the our domesticated animals also. Okay, somatic cell nuclear transfers. Now, nuclear transfer method or somatic cell nuclear transfer, shortly known as SCNT, this is somatic cell nuclear transfer, is a safer and more efficient way to creating the transgene animals in terms of the large number of the embryo being saved. During the SCNT, there is a somatic cell nuclear transfer. A nucleus is taken from the somatic cells, such as a skin cell, other cell like that, then inserted into a transgene by microinjections. Okay, the nucleus is then re-implanted into the unucleated eggs. Okay, from this somatic cell, nucleus is what it is collected, and this nucleus now it is what inserted into a another cell, unucleated cells. Okay, different point. So here in this diagram I have shown you. So from there is animal being, maybe a cattle group. So first is skin cell is collected. Okay, and from here egg is also unfertilized egg is also collected. Okay, now egg is removed with a syringe is shown. Egg is removed, egg nucleus is removed. So the cell become unucleated. Okay, from the skin cell twice the number of the chromosome is collected, and now twice the number of the chromosome is inserted in the egg cells. As a result, the egg cell it attains the what fertilized egg cell, twice the number. Now this fertilized egg cell is allowed to grow. Okay, it develop allowed to grow Okay, once it grow, it will give rise to the blastula. Then again it is implanted, or there is inside the what, a mother. Okay, as a result, it will give rise to a there is a new clone. Okay, or a new that is animal beings. Okay, so which is popularly known as transgenic animals. Okay, and popularly this is known as a cloning also. Okay, so nuclear transfer cloning also. Okay, so again I am repeating this one. The nuclear transfer method, or the somatic cell nuclear transfer, is a safer and a more efficient way to creating the transgenic animals in terms of large number of the embryo being saved. Okay, here the chances of mortality is very less. Okay, during SCNT, a nucleus is taken from the somatic cell, such as the skin cell, then inserted with the transgene by microinjections. Okay, the nucleus is then implanted into the unenculated eggs. Okay, when the egg develops into a blastocyst, it is implanted in the foster mother. Okay, this assures the offspring to be 100% transgenic animals, because the newly implanted nucleus already has the transgene in its genomes. Okay, now here in this picture I have shown the somatic cell nuclear transfer by somatic cell nuclear how transgenic animals are prepared. Okay, so these four methods are very important um, to prepare the transgenic animals. Now, transgenic animals commonly used nowadays. The commonly transgenic animals used are, first is transgenic mice and super mouse, most preferred mammals for experiment. Almost all experimental there is a setup. First, it is done in the mice. Okay, not the mouse. Okay, most preferred mammals for the experiment. Next, transgenic rabbits. It is used for gene farming and molecular farming for drug designing. Okay, the transgenic rabbits. Then transgenic cow. So you might be knowing the Rosie, which is the first transgenic cow which produces the high alpha lactalbumin, milk, high milk protein. This is alpha lactalbumin. Okay, transgenic cow is used. Now, transgenic cattle, increased milk and meat productions. Transgenic cattle are used. Transgenic goat are also used for milk and meat productions. Transgenic sheep, Dolly, you might be knowing Dolly. Okay, the quality and the quantity of the wool productions. Okay, Dolly, first transgenic sheep. Nowadays it is applied in industry, the transgenic sheep cultures. Okay, next is transgenic fishes, rainbow trout, common carp, Atlantic salmon, catfish, goldfish, tilapia, zebrafish, etc. For increase the body growth and size, this was carried out by introducing the growth hormone transgenes. In this fish, if you human growth hormone transgenes you can apply, they will be bigger and larger in size. Okay, and the food value it increases. That is why nowadays it is applied in case of the transgenic fishes also. And lastly, transgenic monkey, ND, transgenic monkey, genetically modified rhesus monkey, first prepared. Okay, so these are the most important transgenic animals used in our society. Now, what are the advantages and disadvantages of these transgenic animals? Okay, though it has got advantage, side by side it has got some side effect also, which is known as the disadvantages. What are the advantages? The advantages of the transgenic animal we can divide into three categories. Some are medical advantages, some are scientific benefits, some are food benefits. Okay, now medical advantages. Transgenic animals can create life-saving medicine of the drugs. Okay, the first. Next, it can produce animals which are stronger and healthier. Okay, construct disease-resistant animals on the humans. The animals serving as a factories for production of the biologically important products are referred as animal bioreactors or farm animals. Okay, they are commercially exploited for the benefit of the mankind. Demand for the organ transplantation, normal physiology, and development is also increasing day by day for what transgenic animals. Okay, so these are the medical advantages. Scientific, what is the scientific benefits? Now they are used for skin the some animals engineered to overexpress the specific protein or knock out a specific protein to help the uncover the newly discovered protein functions. Okay, so there is already I have discussed earlier knockout gene, knock-in genes. Okay, so there is a applied in the transgenic animal, and it is practically it is seen whether the gene is expressing or not, protein is forming or not. Okay, so scientifically it is very important. Now, transgenic animals are used for toxicity safety tests or drug safety tests when drugs are designed. Okay, and toxic whether there is a toxicity or not of the drugs. It is what it is first. It is experimentally done in case of the transgenic animals. Okay, Next is ovulation occurs throughout the year. Another scientific unit, ovulation in the transgenic animal, we can ovulate, okay, throughout the year, so that the ova are readily available for experiment. And it has a shorter breeding cycle. Since it is done artificially, breeding cycle will be very less. Okay. Next, food benefits. Transgenic animals are seen in super fish, super pig, etc., that grow faster and larger and solve the food problems. This has amazing potential to help the solve the world hunger problems. Okay, culturally or in agriculture also, farm animals that can produce the food, milk or efficiently. Okay, efficiently. While so efficiently, while consuming the less food themselves would be another benefit to the mankind. Okay. So in this way, the genetic animals are very important for us. These are the advantages. Now, what are the disadvantages? So, now what are the disadvantages? So, let us discuss now disadvantages. Now, a low survival rate and costly to prepare. The success rate is very low, 1 to 2%. Okay, in case of transgenic animal, mortality rate is very high. Usually leads to the breeding problems. Okay, transgenic animals breeding problems. They face the breeding problems. Now, it leads to the mutation and functional disorders. A new disease can be created on the earth by these transgenic animals, just like a coronavirus. Okay, ethical issues using the transgenic animals. Some ethical issues also there for using the transgenic animals, and the reliability of the transgenic technology is questionable. It might be dangerous to eat transgenic animals. Okay, it may lead to the unregulated expression of the genes. It comes with the possibility of the side effects for some of the animals. Now, the transgenic animals which are used as the bioreactors for the production of the therapeutically important proteins. In industry levels, okay, in agriculture level is used the followings. There is proteins. Okay, for example, first human hemoglobin used from the pig for blood transfusions. Okay, next human lactoferrin in cow's milk promote intestinal iron absorptions. It is also one important that is a protein, human lactoferrins. Now, alpha 1 antitrypsin is in sheep is used to treatment the emphysema. Okay, one the respiratory disorders. SGH in mouse urine is used. One kind of the protein is very important. Human antibodies in mouse administration enhances the immunity. Okay. Next, tissue plasminogen activators in goats used to treating the patients in myocardium in myocardial infection, that is dissolve the blood clot. Okay, human antithrombin 3 in goats regulate the blood clottings. Malaria antigen in goats is used for vaccines. Alpha glucosidase in rabbit treatment for the Pompe disease. So these are the transgenic animals used as bioreactors for the production of therapeutically important proteins. With that, I would like to thank you once again, all of my participants. Okay.